Hello, my name is Patricia Rose De Leon, and today I will be doing the second Talk to the Text assignment for the class Introduction to Human Physiology. So, I believe for this chapter, today we're going to be talking about genetics and DNA function. And yeah, before I actually show you my process of annotation and as well as my thought process, I will look over the different parts or sections of what I will tackle for this subject today. So the first one is will be given the meaning of genetics. The second is the structure and function of the genetic material, DNA replication, structure of DNA. Let's see. Okay. DNA replication, protein synthesis, transcription, translation, protein synthesis, and let's see. Yeah, that will be it. So these are the parts that I need to annotate, and at the end, I believe there is kind of like a worksheet that includes the assignment of codons. That is probably what I can use to memorize the different types of codons. And then we'll move on to amino acids, and I'm not sure what that is, but I think it's for yeah. And then there's also DNA function study questions, which I could also try to answer at the end, near the end of this video. So yeah, since we have a lot to tackle, there's 12 pages of information. I'll just get started right away. So DNA function or genetics. So the first thing that I noticed with this topic is that I've already learned about this topic in my AP biology class, when a class that I took during my junior year. So I'm gonna put a tiny note here in the upper right part, just um, just it just says like look over my AP biology notes, just so I have like these extra information that I can that I already know during my past AP biology class. And it's also a great way to kind of remember all of the information that I've learned in that class and then connect it to this class, to human physiology class. So yeah. So, genetics. Genetics is the science of heredity. The study of genetics includes the study of what genes are, the study of how the genes carry information to the progeny cells. Okay, so I'm not actually aware. Um, I feel like I've heard of what progeny cells are, but I forgot the meaning of it. So I'm gonna highlight it in red, just so I'm reminded to look over that meaning of progeny cells later on, when once I'm done with the annotating process. So genetics is also the study of how genes are replicated, and lastly, it is also the study of how the expression of the information carried in the genes determines the particular characteristics of that organism. Okay, so yeah, those are pretty straightforward definition of genetics. I could understand them clearly, except for the part about progeny cells. Of course, I still need to look that up. But for now, I'm just gonna highlight the meaning of genetics right here. But before I actually move on to the structure and function of genetic material, I'm gonna put a question here at the side because when I was reading this text, I wondered I've wondered a few questions about the function of genes and genetics and how they affect particular organisms such as humans and stuff like that. So my first question is there a genetic difference between races? So this is a pretty general question, and yeah, I'm also wondering about how genetics affects races. Like, is there like a significant genetic difference between the races, or is one or more race, significantly different from the other? And my second question is, from how I interpreted the text, genetics is it's pretty easy to understand when you have like a few fixed outcomes like eye color, but how does that work when it is something like height? So I don't know how to format this in a more appropriate way, but I'm just gonna type it in the same way I said it. When you, when you have a few fixed outcomes like eye color. How do they work when it is something like height? Okay, so I'll just move this right here. And my last question is, do genetics play a part, or do they affect our intelligence, our personality, our habits, or hobbies? If so, how much? So my question is, do genetics affect our... So you might never know, these my questions might actually be answered as I proceed to um, answer the as I proceed to read the text. But some of these are just pretty general questions about genetics as a whole, and yeah, they might be answered by simply googling by simply like searching the answer in Google, or there might be more like objective. But yeah, if if so, how much? So those are my questions for the genetics section of this text. So yeah, so um actually, I think it's a pretty good idea to search this the meaning of progeny cells right now, just so I show you my I'll show you my annotation process, which includes searching for the meaning of unfamiliar words as well. So progeny cells meaning. So progeny, um, probably this is better. So progeny cell is the immediate descendant or descendants of a person, animal, etc. Result of an outcome. Is there a better meaning for it? So basically, a progeny, the organism or the organisms resulting from sexual or asexual reproduction. Oh, okay. So basically, yeah. So progeny. So progeny cells the um are the organisms that result from sexual reproduction. Okay, so now that we're done with genetics, we'll move on to the structure and function of genetic material. So, number one, a gene is a segment of DNA, a sequence of nucleotides in DNA that codes for a functional product, usually a protein. So I'm gonna highlight this section right here. And I'm also gonna highlight nucleotides because I actually know the meaning of what that is, but I'm gonna annotate that later on once I'm done reading all of the 6 parts of this section. So DNA is composed of two strands of nucleotides twisted together to form a double helix. So yeah, um, DNA composed of two strands of nucleotides, and as we know, RNA is only composed of one strand. So each strand of DNA is formed by attaching the sugar of one nucleotide to the phosphate of the next nucleotide. Sugar of one nucleotide to phosphate of nucleotide. So that's information that I need to know of how DNA is formed. The number 4, the two strands of complementary base pairings that are held together by hydrogen bonds between the nitrogenous bases. Adenosine always pairs with thymine, and cytosine always pairs with guanine. The two strands of DNA are thus said to be complementary. Okay, so I'm confused as to what they mean by complementary in this part, and I might have to look that up later. So number 5 is a genotype. Genotype is the genetic makeup of an organism. Keyword makeup. Phenotype, the actual expressed properties such as the eye color or the hair color. The phenotype is the manifestation. Basically, phenotype manifestation of the genotype. Okay, so let's annotate for the first part of this text as well. So we all know what a gene is. It says here it is a segment of DNA, but a gene is composed of sequence of nucleotides. But what is nucleotides? Okay, so nucleotides, from what I remember in my AP biology class, is basically they're molecules that consist of a nitrogen containing base, as the name suggests, such as like thymine or cytosine and DNA, as mentioned here before, and probably cytosine cytosine and RNA. So it is it is a molecule that consists of a nitrogen containing base, as well as a phosphate group and a sugar, which is the deoxyribose in DNA and the ribose in DNA. And RNA, I mean. So, nucleotides are, okay, it is a molecule that consists of a nitrogen containing base, phosphate group, and sugar, which is molecule right here. So DNA is composed of two strands of nucleotides, which means that RNA is only composed of one strand of nucleotide, and that each strand of DNA is formed by attaching the sugar of one nucleotide to the phosphate of the nucleotide, which is connected to what I said right here. Okay, which is connected to what I said right here at the top. So the strands have complementary base pairings that hold together by hydrogen bonds. The adenosine always pairs with thymine. Adenosine and thymine. Okay. Cytosine with guanine. So the true strands of DNA are thus said to be complementary. Actually, before that, it might be helpful if I draw the Lewis structure of adenine and the thymine. So yeah, I'm gonna draw their bond first, just so you know I know what their bond looks like. So I'm gonna draw it right here. Hopefully, I'll have enough space, but that's two bonds, right? This is a double bond. So I did learn this back in my um, AP chemistry class. We did learn about Lewis dot structures, um, geometric shapes of the bonds and stuff like that. So I'm just trying to apply it to this from what I remember. So there should be an H here. There should be a hydrogen bond right here and another hydrogen bond right here. This should be the oxygen bond. There should be a hydrogen bond right here attached to a nitrogen bond. Okay, so that's the adenine and the thymine bond. But for cytosine and guanine, it should be. Wait, it should. I'll just draw it right here. But should I? Do I have enough space to do that? I'll just do it right here, but I'm not sure if I have enough space for that. So I'm gonna draw it right here, but I'm just gonna skip past this video to the final to my final drawing, just so I'm not wasting that much time. So yeah. Okay, so I wasn't able to draw the cytosine and guanine right here in this section because there's not enough space. But if you look over at page 7, I drew the actual guanine and cytosine bond right here because it had enough space. I'm pretty sure that my drawings are not the best. They're not actually. I'm not sure if they're accurate, but I think it's viewable in a way. So okay, so now let's move on to the truth. To this 4C statement that says the two strands of DNA are thus said to be complementary. So I'm not sure what it means by what does it mean by complementary. Does it mean like that the other strand will not function without the other type of meaning? So yeah, I'm not sure what that means, but okay. So genotype, a genetic makeup, an organism. Phenotype, the manifestation of a genotype. Pretty simple definition, pretty understandable. I don't think I need to add any other annotations for that part. Okay, so DNA replication. DNA replication involves the synthesis of duplicate double-stranded DNA molecule. Or in other words, or at least how I interpret it, is basically it's the process by which the DNA makes a copy of itself during the self-division. So I'm going to put that meaning right here. DNA replication, DNA replication, process by which DNA makes a copy of itself during cell division. I'm just gonna put it right here in the bottom. Okay, so B, the structure of DNA. The deoxyribose has carbons numbered from 1 to 5. The phosphate group attaches to the 5 carbon of its nucleotide and to the 3 carbon of the next nucleotide. The nitrogenous base attaches to the 1 carbon. An OH is attached to the 3 carbon, and this is called the 3 end of the DNA strand. The N having a phosphate group attached to the 5 carbon is called the 5 end of the DNA strand. The two strands of DNA double helix are said to be anti-parallel. So anti-parallel because one strand has the chemical sense of direction from 5 to 3 and the other strand has a chemical direction of 3 to 5. So I guess this is the kind of visual representation of that. So before I delve deeper to this visual representation, I feel like I should annotate these different structures of the DNA first. So number one, the deoxyribose has carbons numbered from 1 to 5. Okay, so first off, why why are the carbons numbered from 1 to 5? What is its purpose? What is its the purpose? So yeah, I'm sure yeah, we delve deeper into the structures of the DNA, but I'm not sure what exactly does its purpose serve. Like what exactly does numbering the carbons from 1 to 5 serve? So yeah, the deoxyribose carbons numbered from 1 to 5, and the phosphate group attaches to the 5 carbon of its nucleotide and 3 carbons of the next nucleotide. So basically, the deoxyribose has 5 carbons and 3 carbons. Oh wait, not carbons, not 3 carbons, but rather 3 oxygens. That's the right term. Okay, I don't think there's anything else that we can add into this part. Um, so yeah, it's pretty self-explanatory, and I pretty much understand it. But I'm just gonna move on to this part right here, the DNA nucleotide. So yeah, this is the phosphate group. This is the sugar or the deoxyribose, and this is the nitrogenous base, which I'm not sure. It's not really didn't actually include the structure of it, but I'm just gonna draw it, and it should look something like this. Okay, I think I could do better than that. And there's double bonds here. Okay, I think I did a double bonds right here, double bonds here. So yeah, this is what the nitrogenous base should look like. And looking back, it says here that the phosphate group is attaches to the 5 carbon of its nucleotide and to the 3 carbon of the next nucleotide. So to the 3 carbon. So yeah, so this is the 5 carbon attached to the 3 carbon. I'm so sorry, it's almost 4th of July, so you're probably gonna hear some fireworks in my background once in a while. Okay, so I'm assuming that the nucleotide is referring to the groups in this section, like the phosphate group is a nucleotide, the sugar and deoxyribose is a nucleotide, and the nitrogenous base is a nucleotide. So yeah, got that part, and I'm gonna move on now. So DNA replication. When replication begins, let me just pull up my. So when replication begins, the two strands of parental DNA unwind and separate from each in one small DNA segment after another using an enzyme called helicase. These two separated parental strands of DNA, okay, two two separated strands of DNA, which is the meaning of the helicase, will be used as templates to make two new DNA strands that will be complementary to the template strands. So basically, in other words, I will put this right here. The helicase is, well, in the case of the DNA, they're essential during the DNA replication because they separate double stranded DNA into single strands, which allows each strand to be copied. Yeah, that is the function as well as the importance of the helicase and enzyme. So an enzyme called the DNA polymerase will add new nucleotides to the growing DNA strand. Okay, so in that case, DNA polymerase is used to assemble DNA polymerase assemble nucleotides, or if that's the case, then I could assume that this is probably kind of like the building blocks of DNA. Okay, moving on. However, DNA polymerase can only add new nucleotides. To an existing nucleotide at its tree end. It cannot start the replication of a new strand. Okay, so basically it means that like DNA polymerases they can start from scratch by like adding nucleotides to like a single stranded DNA template. So wait, I think I should just put it here. Cannot start from scratch by adding nucleotides a free single stranded DNA template. Okay, moving on. Um, since DNA polymerase can only add new nucleotides to the tree end of an existing DNA or RNA nucleotide, a short piece of RNA called an RNA primer, starts the synthesis of both of the new DNA strands. So basically, DNA polymerase and DNA polymerase and then RNA primer. They pretty much, what I would say, work together to create the DNA, to form DNA replication, and DNA polymerase, I mean DNA primer is, um, I would say, RNA primer, I would say, is basically the starting point for DNA synthesis. Synthesis. Okay. So the RNA primer is synthesized using um, the enzyme primase. The RNA primer provides, okay, so it doesn't actually give the meaning of the primase, the enzyme primase, which I might have to look up instead. So, function of the enzyme primase function. Okay. So it says here that the primase is an enzyme that synthesizes short RNA sequences called primers. These primers serve as a starting point for DNA synthesis. Since primase produces RNA molecules, the enzyme is a type of RNA polymerase. Oh, okay, got it. So primase. Oh my god, I hate when it starts off with that. So primase, type of RNA polymerase. Is that right? Primase is an enzyme that creates a primer on a DNA strand. Creates a primer on a creates a primer. I'm just gonna put creates a primer. Okay, moving on. The RNA primer provides a tree end to which DNA polymerase can add DNA nucleotides. Um, one new one new D. Okay, go ahead. It's so confusing too. RNA primer tree end to a DNA polymerase. So one new DNA strand called the leading strand is synthesized continuously in a 5 to 3 direction from a template parental strand with a chemical sense of 3 to 5. Okay. So in that case, that would mean that the leading strand is the. Oh, this laptop is so slow. Okay. So the leading strand is kind of like the parent strand of the DNA um because it runs the 3 to 5 direction. Okay, so because it runs the 3 to 5 direction as it says here, and it is, I would say, continuously replicated by the DNA polymerase. So parent strand of the DNA runs in the 3 to 5 direction and is continue continuously replicated by DNA polymerase. Okay, so we're done with wait, I don't think we're done with the part. Okay, so there's 7 more parts to this, and yeah, let's just get started. So number 8, the leading strand has only one primer and is made continuously because it has three end at the replication fork to which DNA polymerase can add new nucleotides. Okay, so the leading strand has only one primer and is made continuously because it has three end at the replication fork. So the other new DNA strand is called the lagging strand and is synthesized discontinually in fragments of about 1,000 nucleotides called Okazaki fragments. Okazaki fragments. The lagging strand has the chemical sense of 3 to 5 and the template strand has the chemical sense of 5 to 3. The lagging strand will have many primers and the replication of this strand will move in the opposite direction of the replication fork. Since it has a 5' at the replication fork and a DNA polymerase cannot add nucleotides to a 5'. The RNA nucleotides of the RNA primer are later digested away by the 5 to 3 exonuclease activity of the DNA polymerase. So the lagging strand will have many primers in the replication of the strand and will move in the opposite direction since it has a 5. The DNA polymerase cannot add 20%. So DNA nucleotides replace the RNA nucleotides, and DNA ligase joins the nucleotides together to make the continuous strand. The replication fork is the point at which replication is occurring. Each new double-strand DNA molecule contains one original strand conserved and one new strand. Thus, the process of replication is called semi-conservative replication. Process of replication is called the semi-conservative replication. Okay, so I moved forward and basically highlighted all of the important terms that I encountered in this text, and because most of the meanings, at least, they were pretty understandable, so I didn't need to put any annotations with their meanings, and I think it's pretty self-explanatory when you just read the text overall. But before I move on to the protein synthesis, I have a few questions about DNA replication and all. So number one, in DNA replication, what is meant by 5 to 3 orientation and vice versa? So this is something that has been pretty confusing for me since I was reading this text, and I think I need a bit of like clear up with what that actually means. But I do have a few questions about DNA replication and its process in, overall. So why do my first question is why do mutations occur at all during DNA replication, and how or why do the proteins that replicate DNA mess up? And to follow up that question, my last question would be is if DNA replication couldn't make any errors, would species evolution be possible? So yes, those are a few of my questions, inquiries about my final questions for DNA replication as a whole. So I'll move on to the protein synthesis now, and yeah. So number one, the proteins of the cell will control its function and structure. The proteins of the cell will control its function and structure. DNA controls what the cells do by dictating which proteins are to be made, by the cells. The DNA contains the instruction. Keyword contains the instruction for the synthesis of the proteins. A gene is a segment of DNA or a sequence of nucleotides in the DNA that codes for the functional product, usually a protein. The DNA molecule doesn't directly participate in the assembly of the amino acids into the proteins. However, the DNA does is give the instructions to the mRNA. So I forgot what the mRNA is actually is. I'm sure that I've tackled it before in AP biology, but I think I need to cover that a little bit more. So I think I will just write this in red. Each protein has a specific order in which the amino acids are linked. Just one change in the amino acid sequence will change the nature of the protein. So the DNA specifies the correct sequence of amino acids in the formation of the protein. Okay, keyword. Each protein has a specific order in which amino acids are linked. Just one change in. Okay. It is the mRNA which carries. Okay, so I don't need to actually. Um, I don't need to look this up because this is actually already mentioned in the text. So basically, RNA is something that carries specific amino acid sequence from the DNA molecule to the site of the protein synthesis, the ribosomes. So in order for the proteins to be made, first the mRNA must be transcribed from the DNA molecule. So protein synthesis in summary is okay, so protein synthesis in summary is the process by which cells make the proteins. So yeah, process protein synthesis process by which the cells make proteins. So it occurs during two stages which was mentioned in this text, which was um the transcription yeah, I can the transcription and then the translation. So overall, I think I understand what protein synthesis is. I basically highlighted all of the important, at least the key information that I need to know about protein synthesis. So, in summary, protein. So, why is it important? Why is protein synthesis important? So, it is important because it is an essential process that happens regularly within the cells. And it is used to create new proteins that are used for various processes in the body, in the DNA, and stuff like that. So, yeah. So I think it's safe for me to move on because I think this section was pretty short, and I pretty much understand everything. So there's really not much anything else needed to be added on. So let's move on to transcription. So transcription is the synthesis of complementary strand of RNA from a DNA template. So transcription requires an enzyme called the RNA polymerase and the supply of RNA nucleotides. Only one strand of the two DNAs serve as the template for the RNA synthesis. This is called the antisense DNA strand. The complementary DNA strand is called the sense DNA strand. Okay, so we still have a bit of information to go through, so I'll try to go over this pretty quickly. The prompter, and the terminator sites when a DNA molecule is involved in the transcription. The prompter site is in the region in the DNA where RNA polymerase initially binds and where transcription begins. The terminator site is the region of the DNA that signals the endpoint for the transcription. And C, the base sequence that specifies the prompter or the terminator sites on the DNA is not transcribed or translated. Transcription begins when RNA polymerase binds to the prompter site. polymerase to the prompter site and keyword begins. The hydrogen bonds between the two strands of DNA are broken so that the portion of the DNA molecule unwinds and separates. Oh my god. Mm, to, um, okay, I might have to pause for you right now. Okay, so sorry about that. I think my Wi-Fi went out for just a few seconds, but let's continue on. Okay, so RNA is synthesized to the in the 5 to 2 direction. Three RNA nucleotides pair up complementary to the nucleotides in the DNA template strand. Opposite A on the DNA will be U on the RNA. Opposite T on the DNA will be A on the RNA, and opposite G on the DNA will be C on the RNA. Opposite C in DNA will be G on the RNA. Okay, let's just stop right here, and I think I need to highlight this whole section right here because um that is pretty confusing to understand at first. I mean, I might get it um once I search it up, but I'm just gonna put a tiny note here that says I'm confused at this part. Look up for further explanation. So yeah, that is just a reminder for me to look up what this means. So the enzyme RNA polymerase links to the RNA nucleotides together to form the RNA strand. When the RNA polymerase reaches the terminator site, the transcription is complete. Ribosomal RNA is transcribed in the nucleolus and messenger, and transfer RNA are transcribed in other parts of the nucleus. So ribosomal RNA, nucleus and messenger, and transfer RNA in other parts of the nucleus. Okay, a few things here. So basically, in summary, transcription is the um. Okay, basically transcription is like the process by which the information in a strand of DNA is copied into a new molecule of messenger. Messenger RNA, or also called as mRNA. mRNA. So yeah. So during this process, I will just put this right here. So during this process, basically the DNA is safely and stably stores genetic material in the nuclei of the cells as reference or template. And it is also in this phase, meanwhile, mRNA is, kind of, I would say, like kind of like a reference book because it carries the information as DNA, but it is not really that used for like long-term storage, and it can freely exit the nucleus, so it is used for long-term storage. So although our mRNA contains like the same information, I don't think it's the same um It is not like an identical copy of the DNA segment because the sequence is like complementary to the DNA template, as mentioned before. So mRNA is not an identical copy of the DNA because the sequence is complementary complementary to the DNA template. Okay. So yeah, basically transcription is like being carried out by an enzyme called the RNA polymerase and a number of like accessory proteins, as mentioned, kind of like the transcription factors. And these transcription factors, they're used to bind to like specific DNA sequences, in order to kind of like recruit the RNA polymerase to like an appropriate transcription site. And yeah, okay, so um one question so far for this segment, or at least from following after this segment, after reading this segment, is how are genes, DNA, RNA, and mRNA, how are they all connected? So since I'm studying this right now, and yeah, I think this part totally got me confused, and I think I need some clarifications. Like, what does a gene have to do with proteins? How do they form? Just, I feel like I need a little bit of elaboration on that part. Okay, so we're done with the transcription, and all I need to go over is the translation and the last part of protein synthesis. So translation is the process in which the mRNA dictates the synthesis of a protein on the ribosome. And there are four types of RNA which play a role in protein synthesis: the precursor messenger, pre-mRNA, messenger RNA or mRNA, transfer RNA, and ribosomal RNA. So the precursor messenger is transcribed and is then altered in the nucleus, transcribed and alkylated in the nucleus to form the messenger RNA. So, four things about the precursor RNA. The precursor messenger RNA is larger than the mRNA. The pre-mRNA contains introns, which are non-coding DNA within a gene, and exons, which are regions of the gene that code for the protein. So I'm not gonna add other information for that. It's pretty straightforward. So the introns are cut out, the pre-mRNA and the exons are spliced together by the molecules called the small nuclear ribno ribnucleoproteins, or SNRNPs, or SNRPs. SNRPs are small ribosome-like aggregates of RNA and protein that form a body called a splicosome that splices the exons together. Actually, that splices the exons together to form the messenger RNA. So the messenger RNA carries the information from the DNA to the ribosome, the orders specifying the orders of the amino acids in the proteins. Each amino acid is coded for by three nucleotides called a codon. Sense codons are codes for amino acids. Nonsense codons are stop codons, do not code for amino acids. Okay, pretty self-explanatory. Transfer RNA, or tRNA, picks up the amino acids from the cytoplasm and transports these to the amino acids to the ribosomes, so they can be assembled into into protein. The tRNA molecule is a single-stranded nucleotide sequence which is shaped into a clover leaf configuration. So an amino acid is attached at one end of the tRNA molecule by an enzyme called the amino acyl synthetase. Oh my god, synthetase. Which amino acid is attached to a tRNA molecule is determined by the three nucleotide sequence found at the other end of the tRNA molecule. This three nucleotide sequence is called the anticodon. Sorry about that. So let's just keep going. It's called the anticodon. The anticodon is complementary to the codon of the mRNA, and there is at least one tRNA molecule for each amino acid. Some amino acids have more than one codon. Therefore, some amino acids have more than one tRNA because the anticodons are different. 6. Ribosomal RNA are proteins that form the ribosomes. And ribosomes are the sites. Okay. Form the ribosome. Ribosomes are the sites where the amino acids are joined together by peptide bonds to form a protein. Ribosomes are made up of RNA and proteins, and there are two subunits making up a ribosome, a larger 60S subunit and a smaller 6040S subunit. The 60S subunit has three binding sites for the tRNA molecules: the E, the A or amino acyl site, the P or the peptidyl site, and the E site. The 60S subunit also contains the peptidyl transferase, which forms the peptide bonds between the amino acids forming the protein. The 4S subunit binds the RNA molecules to the ribosome. The RNA interference. The RNA interference is a regulatory process performed by the RNA molecules that don't code for proteins but may prevent the mRNA molecules that do code for the proteins from being translated into those proteins. Andrew Fire and Craig Miller won the Nobel Prize in Physiology and Medicine in 2006 for the discovery of a double-stranded RNA molecules that play a role in RNA interference. So Andrew Fire and Craig Miller did that. So RNA interference is mediated by two types of is mediated by two types of double-stranded RNA molecules: short interfering RNA or CIRNA and the microRNA or miRNA. Only of the two RNA strands of the I'm just gonna pronounce them in a way it's more easier, but I know what they actually mean, so it doesn't matter. So the two RNA strands of the serina and the miRNA enters a protein particle called the RNA induced silencing complex that single strand can be complementary with a protein coding mRNA and that interfere with the synthesis of protein. In the future, RNA interference may be used to medically suppress the expression of specific genes that may cause disease. Okay, so that was a lot of information, and because I don't want to keep this video a bit longer, I'm just gonna move forward to when I put all of my annotations for this part. Okay, so I'm done putting in the annotations for translation, and I'm just gonna tell you the annotations that I put down for this section. So basically, first I just summarize what translation is and how I understood it. Is basically translation is the process of translating the sequence of the messenger or the RNA or mRNA molecule to a sequence of amino acids during the protein synthesis. And the pre-mRNA requires a kind of splicing or the removal of introns to produce the final mRNA in the molecule containing only exons. And the mRNA is this single-stranded molecule that carries genetic code from DNA in the cell's nucleus to the ribosomes, the cell's protein-making machinery, as mentioned before. And the transfer ribonucleic acids, or the tRNA, is a type of RNA molecule that helps decode a messenger RNA or mRNA sequence into a protein. And the primary function um, of an R of a ribosomal RNA is in protein synthesis, which is to bind the mRNA and the tRNA to ensure that the codon sequence of the mRNA is accurately translated into the protein. And so the last part is the RNA interference. I pretty much understood what it is, but I kind of included one of the reasons why it is important, especially when this last statement was made. So basically, RNA interference has this important role in defending cells against parasitic nucleotide sequences, either viruses or transposomes, and it also influences development. So yeah, that is all for protein synthesis, translation, and transcription. So I guess we can move on to um, protein synthesis, the last part, um, at least for this section. And yeah, I might still have to check this out, but okay. So let's just continue. So the protein synthesis in the mRNA strand contains the information from the gene to synthesize a protein. Um, oh, is this just a summary of what we went over? Okay, so the mRNA strand contains the information from the gene to synthesize the protein. At the beginning of the mRNA strand is the start codon, which is always AUG. The start codon tells the ribosome to start assembling the amino acids into the protein. At the end of the mRNA strand is the stop codon. Basically, it's in the name, it's to tell the ribosome that the protein is complete. If the protein is to be made, if the protein to be made is 3 amino acids long and consists of the amino acids alanine, glutamine, and leucine, the mRNA strand will be AUG, GCU, CAA, CUU, and UAA. Start alanine, glutamine, leucine, and stop. Okay. So start will always be AUG in this case in photosynthesis, and stop will always be, I guess, a UAA. Protein synthesis begins when the ribosomes move into the when the ribosomes move onto the mRNA strands. Okay, why not? Let's just highlight that whole part. So the ribosome moves across the mRNA into the 5 to 3 direction. The anticodon must be complementary to the codon in order for the tRNA to bind to the ribosome. This assures that the correct amino acid is placed in the position dictated by the DNA. The enzyme peptidyl transferase joins the amino acids to form the peptide bond. Form the peptide bond. When when the ribosomes move on to the stop codon UAA, the ribosome knows that the protein is complete. Post-translational processing is the process of the polypeptide chain. Once the polypeptide has been made by the ribosome, it undergoes a modification. The, the methionine coded by the start codon is removed by the enzymatic action, and the final polypeptide chain is as follows: ALA, GLN, and LE. U. Therefore, it is the gene on the DNA which dictates the proteins to be made on the ribosomes. The DNA sends its messenger, mRNA, to the ribosomes in the cytoplasm, and the amino acids are assembled in a specific order depending on the sequence of codons on the mRNA. The tRNA molecules bring the specific amino acids to the ribosomes to be assembled into the protein. Only the tRNA molecules whose anticodons must match the codons on the mRNA will attach to the ribosomes. Thus, only the amino acid sequence dictated by the DNA is formed. The DNA controls cellular functions by dictating the synthesis of proteins by the cell. These proteins will be structural proteins or enzymes which control the cellular functions such as controlling the transport of molecules into and out of the cell. Some membrane proteins are controlling cellular chemical reactions. So this part right here is just a summary. I don't think I need to add on anything to it. It's basically information that I already went over, and in that sense already put in some annotations. So I won't be adding in annotation for this part since it's just a summary. Okay, so I will now move on to the assignment of codons. Before I actually try to interpret this, I will read the information that is down here. So, for example, the codon for phenylalanine is UU and UUC. Notice that each codon is specific for amino acids. Conversely, an amino acids can have several codons. For example, phenylalanine, oh my god, phenny phenylalanine, as shown above. Having more than one codon per amino acid helps to decrease problems with putting the wrong nucleotide in the mRNA chain. Notice that any codon beginning with the GU will be valine. Also notice that the UAA and the UAG and UGA signal the end of a protein are nonsense or stop codons that do not code for the amino acid. Okay, I guess these are a few examples of the codons. Okay, so the assignment of codons, first position, U. In the second position, U will be, it will consist of fe and lu. And then C, it's sir. A is tier and stop. And G is cis, stop, and TRP. And in the third position, it's UCAG. In C, U is Liu, C is pro, A is his or GLN, and G is ARG, and the third position is UCAG. Probably that's the position for all of them. But A is in the second position, IL or ILE, MET star, THR, ASN, and CER. And then G, Val, um, um, in the second position is Val, ALA, ASP, and GLE. Okay, so I think this is pretty much how the meaning behind those words. So basically, these are amino acids. ALA is alanine, or ARG is arginine, ASN is asparagine, ASP is aspartic acid, cis is cysteine, GLN is glutamine, GLU is glutamic acid, GLY is glycine, his is histidine, ILE is isoleucine, LEU is leucine, LYS is lysine, MET is methionine, PHE is phenylalanine, Pro is proline, SER is serine, THR is threonine, TRP is tryptophan, TYR is tyrosine, VAL is valine. So I think this is pretty much things that needs to be memorized. So I'm gonna put this up right here. Oh my god, it's so hard to write down what mouse. Okay, I guess so. I guess these are just a few examples of the the codon as well. Yeah. So these are the DNA functions and study questions. I guess I could separate them from my annotation process. I mean, yeah, I could just treat them as a quiz, and I probably don't need to show my process for this one. It's just review, so I won't be adding any annotations for that. But anyways, so I guess that pretty much adds or concludes all of the topics encountered for um, reading aloud second assignment, which focuses on the central dogma by Mizuno. So yeah, um, I hope you enjoy watching me expressing my thought process as well as my annotation process for this text. And as always, this video might be a bit too long for Canvas to upload, so instead the I will add the Google Drive link to the comment section of the submission box in Canvas, and there you will be able to see two parts of the video. The first one is the beginning, and the second one will be this one, which is the last one. So yeah, thank you so much for listening, and have a nice day.